To the public, epigenetics is known as a more natural way of tampering with the observable traits. The process of epigenetics happens naturally. By definition, it is the change in the observable traits, also known as a phenotype, without the change in the inheritable genetic makeup, otherwise known as the genotype.
The topic of epigenetics began with the biologist named Conrad Hall Waddington. He is renowned as the founder of epigenetics, although he was not searching to be. He was researching a broad topic of combining genetics and developmental biology.
Moving forward about 28 years, the most commonly used method of epigenetic mechanism change is DNA methylation. It is the process of adding a methyl group to the 5 carbon on the cytosine, resulting in methylcytosine. The methylcytosine thereby affects the gene expression during transcription, thus making it a form of transcriptional control mechanism. An example of post-transcriptional epigenetic change is histone modification. Histones are proteins that order DNA into structural units called nucleosome. They furthermore play an extremely important role in transcription repression or activation. The modification can happen in the form of methylation, phosphorylation, or acetylation. Each induces replacing a hydrogen with methyl, phosphate, or acetyl using substitution reaction. Because of the new methyl, phosphate, or acetyl group, the newly translated gene is not expressed or expressed differently due to the peptide chain being altered.
In addition, when the acetyl group is added, it helps to regulate epigenetic gene expression by affecting cellular processes including transcription, gene slicing, cell cycle, apoptosis, DNA replication, and DNA repair. Furthermore, adding an acetyl to histones loosens their association with DNA, exposing the promoter. Some techniques that are involved to study epigenetics that carry out its procedures includes bisulfite sequencing, which is a method that identifies sites of DNA methylation. To begin the process, bisulfite changes cytosine to uracil, however it leaves 5 methylcytosine residues unaffected. Another technique that is used is called sequencing. This method helps to recognize methylation pattern and allows to measure its percentage. Pyrophosphate release and the creation of light on a nucleotide is detected to measure the percentage of the target sequence. Furthermore, through the use of PCR, direct fragments can be used for this process without the use of cloning.
But the entire reason why epigenetics is being widely funded and followed by scientists is because of the variety of illnesses like cancer showing epigenetic mechanisms behind them that could potentially be stopped if further research was done into epigenetics. Cancers in particular suppress specific cancer silencing genes through epigenetic mechanisms. By finding a way to stop these from happening, we can be one step closer to finding a cure for cancer and many other illnesses that use epigenetic methods. Furthermore, there are more positives to researching and funding epigenetics for the betterment of mankind.
Epigenetics provides an evolutionary advantage to society and deploys a tool that senses challenges and primes genes to efficiently respond to similar threats in the future. It is important to study epigenetics because it is linked to several mutations. In addition, mutations happen randomly due to environmental factors, or they are epigenetic. Several diseases and symptoms are linked to epigenetics, which is why it is beneficial to further study it to help us find cures for certain illnesses. Furthermore, studies show that epigenetics effects occurs over the full course of a human lifespan, unlike other genetic changes that can only affect humans as babies. Even though all the factors posed against epigenetics, the rapid increase in popularity will cause a revolution in bioscience.
Although epigenetics has been rising in popularity rapidly, the rise in popularity could be equally as bad as it is good. As seen from other now controversial topics such as neuroscience or stem cells, the preliminary findings or pioneer scientists did not notice the consequences for human health or well-being. Claims against neuroscience and stem cells was originally made by policymakers, social scientists, and media commentators.
In conclusion, epigenetics has many pros and cons linked to its more natural methods of controlling gene expression. This subject is still in its infant stage and needs to be thoroughly researched before entire conclusions can be made. We hope you enjoyed our video and hope you learned something new. Thank you for watching.